Life happens fast, really fast. At any given time, cells of all kinds carry out thousands of metabolic reactions, breaking down some molecules and building others. And all this has to happen fast enough to meet cellular needs. A bacterial cell, for example, may have about 20 minutes to carry out all of its life's activities, including copying all of its DNA before it divides. So it's pretty clear that if any of the necessary chemical reactions take too long, there's going to be a major problem. It so happens there is just such a reaction. It's one involved in making a crucial component needed for DNA. It takes 78 million years or so. What's a bacterium to do? Well, find a way to speed up the reaction, that's what. But what sort of wizardry can speed up a reaction that takes 78 million years so much that a bacterium can finish out its life activities in 20 minutes? Prepare to be impressed. The agent of this dizzying acceleration is a biological catalyst called an enzyme. OMP decarboxylase, to be precise. And what does this enzyme do? It speeds up the reaction, so it's done in less than 20 milliseconds. That's a speed up of 140,000 trillion times. Yep. And it's not just bacteria. All organisms need and use this enzyme to catalyze synthesis of pyrimidine nucleotides for making RNA and DNA. A reaction that without the enzyme takes millions of years to complete can be speeded up so it's done in milliseconds. That's the power of enzymes. Ordinary chemical reactions would happen far too slowly to support life. This is biochemistry, where reactions happen at the speed of life. So what are enzymes and how do they accelerate chemical reactions to such an extraordinary extent? Enzymes are catalysts and catalysts speed up reactions, but they're not changed by the reactions they facilitate. Not all catalysts are found in cells. A familiar non-biological example of a catalyst is the catalytic converter in cars. It's called a converter because it converts pollutants like carbon monoxide and nitrogen oxides into less harmful substances. It's called catalytic for two reasons. First, it increases the rate at which polluting molecules are transformed into more benign compounds. And second, under normal conditions, the catalytic converter itself can be used over and over without being changed by the process. Enzymes are called catalytic for the same two reasons. They speed up the rate at which biochemical reactions happen in cells. And each enzyme molecule can be used over and over without being changed. Now, enzymes are proteins just like other proteins we've seen. They contain amino acids whose unique side chains play important roles in the mechanisms of catalysis. But what makes enzymes truly amazing catalysts is their efficiency. The metal catalyst in a car's catalytic converter might speed up a reaction by factors of 100 or maybe 1,000, but enzyme catalysts speed up a reaction by 1 billion times, like the sugar metabolism enzyme triose phosphate isomerase. Often the speed up is even more mind-boggling amounts, as we saw for OMP decarboxylase. It's not uncommon for enzymes to speed reaction rates by a factor of 1 trillion or more, as happens with adenosine deaminase, which affects synthesis of nucleotides needed for making DNA and RNA. Another remarkable thing about enzyme catalysis is the fact that it happens at the temperature and pressure conditions of living cells. By contrast, consider the conditions needed in factories to synthetically make something as simple as ammonia. Ammonia, or NH3, is a very simple molecule made up of 1 atom of nitrogen and 3 atoms of hydrogen. A widely used industrial process to make it combines nitrogen from the air with hydrogen in the presence of a metal catalyst. But they form ammonia only if heated to 450 degrees Celsius. That's 840 degrees Fahrenheit, at 200 times atmospheric pressure. Now compare that with the so-called nitrogen-fixing bacteria. These bacterial cells make ammonia from nitrogen in the air far more efficiently without the need for high temperature and pressure, all because they use enzymes as catalysts. Clearly, enzymes can do remarkable things. Before we investigate how enzymes do what they do, some simple terminology. The molecule or molecules being acted upon by an enzyme are called substrates. In the reaction for making ammonia, nitrogen and hydrogen are the substrates. Substrates are also called reactants because they are the molecules undergoing the reaction. The molecule or molecules produced by the reaction are called products, not surprisingly. In our example, we had only one product, ammonia, but some reactions have multiple products. Most enzymes have names that end with ase or ase. When you encounter a molecule whose name ends in ase, you can tell that it's an enzyme. The few examples that are exceptions include some of the first discovered enzymes for digesting protein like pepsin or trypsin and chymotrypsin, but that's not common. The rest of an enzyme's name tells something about what the enzyme does. Sometimes the name tells you which substrate the enzyme breaks down. For example, a lipase breaks down a fat. Lactase breaks down lactose. A protease breaks down a protein. Other enzyme names indicate an output or product. ATP synthase, for example, is the enzyme that catalyzes synthesis of ATP. Many other enzymes have names that describe a type of reaction using words like synthetase, oxidase, reductase, transferase, or isomerase. In general terms, we can describe any reaction involving enzymes as substrate S being converted to product P. Now let's look at what's happening in catalysis. For this, we use something called the Gibbs free energy, written simply as G. It's a measure of the amount of energy available to do useful work in a process. In a biochemical reaction, the work being done is making and/or breaking of chemical bonds. We can follow the energy changes for a reaction in a simple graph. On the y-axis, we see the Gibbs free energy of the substrate and the product of the reaction. And on the x-axis, we are looking at the progression of the reaction. On the far left, we see the free energy of the substrate, the glucose 1-phosphate, or G1P. On the far right, we can see the free energy of the product, G6P, or glucose 6-phosphate. The green line tracks the progress of the reaction from an energy perspective. First, we see the uncatalyzed reaction. Now, here are some of the most important things to pay attention to. First, the free energy of the final product is lower than the free energy of the substrate. The difference between these energies is the change in Gibbs free energy during the reaction. Now, the Greek letter delta is commonly used to denote change, so this change in free energy is called delta G. The net change in free energy, or delta G, is negative in the conversion of the S to P, or the substrate to product. This means that energy is released by the reaction when S is transformed to P. You will note, though, that at first the green line goes up from the energy level of the substrate, indicating that some energy input is needed before the reaction proceeds downhill. The difference between the energy of the substrate and the peak of the line is something we call the activation energy for the reaction. Now, you might be wondering why activation energy is even needed. If the final product is at a lower energy than the substrate that you started with, why would you need to add any energy for the reaction to happen? To understand this, we need to think about what a chemical transformation really is at the molecular level. To convert one or more substrate molecules into a product, some bonds have to be broken and new ones have to be made. For example, the substrate molecule or molecules might have to be forced or bent into a form that will allow existing bonds to break or form, just as you might need to bend a stick to weaken it at the spot you want to break it. This contorted form of the reactants is called the transition state, and to reach it takes energy, just as you would need to put in effort to bend that stick. That's what the activation energy is, the energy needed to get molecules to that transition state. The input of the activation energy is why the transition state is the highest energy level on the graph. Now, the transition state is a short-lived and unstable state, just as a bent stick does not stay that way for long. It either breaks or returns to its original state. Once the substrate molecules are in the transition state, conditions are favorable for the original bonds to break and new ones to be made. But where does the activation energy to get to the transition state come from in the first place? Well, under normal conditions, molecules are in constant motion, driven by thermal energy. They bounce around, they bump into each other. A particularly energetic jostle may send some of them into a state of higher energy. So at that point, they're at the highest point of the energy curve, the transition state, from which they can then go downhill easily. This happens in the absence of catalysts, but rarely. Things can be speeded up a bit by raising the temperature, but the input of heat energy makes molecules move faster and bounce around more. But to make a really significant change in the rate of a reaction, cells need the assist of a catalyst, since the range of temperatures they can survive at is pretty narrow. Now let's take a look at the same reaction catalyzed by the enzyme phosphoglucomutase. Here I've used dotted lines to show where the energies differ from the uncatalyzed reaction. And we see that the activation energy in the presence of the enzyme is significantly lower, but neither the beginning Gibbs free energy nor the ending Gibbs free energy was altered. Thus, the energy released in going from the substrate to the product is the same, regardless of whether or not the enzyme was involved. So the only thing that has changed is the activation energy. Put another way, the catalyst has reduced the activation energy it takes to get the substrate into the transition state. When the activation energy is lower, many more substrate molecules reach the transition state at a given temperature, so the conversion of substrate to product is correspondingly faster. For a reaction to happen, reactants must be brought into a transition state that allows those bonds to be broken so new ones can be made. This requires activation energy. In the presence of enzymes, the amount of activation energy needed is reduced compared to the uncatalyzed reaction. So how does the enzyme make it easier to reach the transition state? Well, enzymes have an active site, which is a pocket into which the substrate fits and where the chemical reaction occurs. The pocket is created by the 3D folding of the polypeptide. Amino acid side chains protrude into the cavity of the active site, giving it its very specific shape, with chemical characteristics that derive from their side groups, as we've learned before. Each will be positioned at the specific places in the active site, ensuring that only substrates with the appropriate surface characteristics will be able to bind. The high specificity of substrate binding sites by enzymes gave rise to a simple but not entirely accurate analogy that a substrate fits an enzyme like a key fits a lock. In this lock and key model, called the Fisher model, the enzyme is envisioned as an exact complement to the shape the substrate it acts on. But for most enzymes, an induced fit model is a better description of what happens. In it, the binding of the substrate at the active site changes the enzyme-substrate combination transiently, making the two of them fit closer together. This involves a slight alteration of the shapes of the enzyme, which is why flexibility is important, and the substrate, a bit like inserting your foot into a shoe that is not the shape of your foot. The footwear stretches, and your toes might squish a bit, resulting in a snug fit. An enzyme's flexibility lends itself to several catalytic mechanisms. One mechanism of enzymes is to bind the substrate in a way that provides a shortcut to the transition state. That is, the best fit of the enzyme for the substrate favors the transition state where bonds can be broken and new ones made. Blocking the normal activity of such enzymes is the idea behind a class of drugs called transition state analogs. Another mechanism, if there's more than one substrate, is as a juxtaposer that puts molecules into close proximity. Outside of the enzyme, the molecules depend on random molecular collisions to bump into each other. There are many unproductive collisions, while useful interactions are rare. But inside the active site, the substrates are closer and optimally positioned to react with each other. Now consider hexokinase. It's an enzyme that's involved in the breakdown of sugars. Its structure resembles a pair of jaws, just like I have my hands. The two substrates are glucose and ATP. And the binding of the substrates causes the jaws to clamp down, squeezing them together, favoring the transfer of a phosphate from ATP up here to glucose down here. So in this reaction that I've just described to you, we write it as glucose plus ATP yields, and that's what that little equal sign is in the middle, glucose 6-phosphate plus ADP. And that's what you've just seen happen with my hands here. When the reaction is done, the jaws open and the products are released. That's the glucose 6-phosphate and the ADP. Enzymes commonly use other molecules to help in catalysis. Metal ions such as iron, zinc, and copper serve as cofactors that aid reactions. In other cases, bigger molecules called coenzymes help to move electrons, atoms, or entire functional groups. In oxidation, a very important example is the electron-carrying molecule NAD+, which is partly composed of the B vitamin niacin. Electrons lost by reactants during oxidation are absorbed by NAD+. Several other B vitamins are precursors for coenzymes that greatly expand the type of reactions that enzymes can perform. Some enzymes directly participate in the reaction they are catalyzing, usually by the enzyme's amino acid side chains, forming transient bonds with the substrate. Now, a well-studied example illustrating this mechanism is the digestive enzyme chymotrypsin, which chops up proteins we eat by breaking the peptide bonds that hold their amino acids together. Chymotrypsin breaks peptide bonds adjacent to amino acids with large aromatic rings, such as phenylalanine, tyrosine, and tryptophan. When chymotrypsin binds to a protein substrate, that triggers a change in the amino acid side chains in the enzyme's active site. That's the first step in the catalytic process that results in the breakage of the peptide bond within the bound protein. Now, zooming in, chymotrypsin's active site has 3 amino acid side chains extending into the active site. Known as the catalytic triad, these belong to the amino acids serine, histidine, and aspartate. Now, binding of the proper substrate squeezes them closer together. The negative charge of the aspartate side chain essentially pushes electrons in the histidine ring towards the hydroxyl or OH group on the serine side chain. These negatively charged histidine electrons pull a positively charged hydrogen or proton off of the serine's OH hydroxyl group, leaving behind a negatively charged oxygen called an alkoxide ion. All of this chemistry happens in the side chains of the catalytic triad. Now, why is this relevant? Well, the negatively charged alkoxide ion formed on serine is very reactive, and it reacts with the nearest chemical bond to it, which happens to be the peptide bond of the bound protein, adjacent to the phenylalanine in the substrate binding site. Voila! The peptide bond is broken. Though there are other steps to the process, the enzyme has essentially done its thing at this point, thanks to the small changes induced in the enzyme by the binding of the substrate. At completion, the enzyme releases 2 pieces of what had previously been a single polypeptide chain, and the catalytic triad returns to its original state. Enzymatic reactions do not occur in isolation. In cells, the vast majority of them are interdependent, with the product of one reaction serving as the substrate for the next one, like a factory's assembly line. Production of biomolecules depends on chains of reactions, each fed by the one before it, each catalyzed by a specific enzyme. Now, in thinking about enzymes, let's start with a real-world analogy to help understand some parameters of enzyme activity. We can compare enzymes to workers in a garment factory, for instance. Workers need raw materials, or substrate, to make their products. Fabric is the substrate here, and workers are the enzymes. If a factory has plenty of workers working hard, the primary limitation for maximizing the number of garments produced in a day is having plenty of fabric. If workers have to wait for fabric, time will be wasted and the maximum number of garments will not be reached. If fabric is not a limit, then the factory will reach its maximum number of garments per day, but no more. Providing additional fabric will not result in production of any more garments because the factory is at capacity. Enzyme velocities are measured in product made per time, just as the factory measures productivity by the number of garments produced per day. If a substrate is in short supply, the enzyme will spend time idling and maximum velocity will not be reached. However, if the enzyme is saturated with substrate, meaning that as soon as a product molecule is released, a new substrate is bound, then the reaction will reach a maximum rate or velocity. For an enzyme, we call this quantity Vmax for maximum velocity. Now let's imagine the factory is making a popular garment, and as soon as one is made, it gets snatched up by a buyer. Well, when that happens, the company decides to expand production and does so the only way possible, by adding more workers. If the company was making 500 garments per day with its workers, then added enough workers to triple its workforce, the new rate of production would increase to 1,500 per day. The maximum production is set by the number of workers who turn fabric into a garment, assuming there's no shortage of fabric. Now, likewise, in the case of an enzyme, maximum velocity is set by the number of enzyme molecules performing catalysis. If we have twice as many enzyme molecules, the Vmax will be twice as large, so long as there's plenty of substrate for conversion into product. Vmax tells us about the activity of an enzyme, but it is not entirely useful for comparing different enzymes. In the factory, for example, talking only about the number of garments made per day is not enough to compare the productivity unless you knew how many workers each factory employed. To make such comparisons more meaningful, biochemists use a measure called the catalytic constant, or kcat. It's also called the turnover number of the enzyme. Now, kcat is useful because it takes into consideration the concentration of the enzyme, that is, the number of workers in the factory analogy, as well as the maximum velocity, which is total production. So the turnover number kcat is simply the Vmax, the maximum velocity, divided by the concentration of enzyme used, which essentially gives the number of product molecules made per enzyme molecule per second. Okay? That's equivalent to the number of garments made per worker per day, which you understood. This, as you might imagine, is a better way to gauge productivity than the total number of garments made or the total amount of product made. Now, some enzymes have kcat turnover numbers that are absolutely incredible. The enzyme carbonic anhydrase, for example, catalyzes the formation of carbonic acid from carbon dioxide and water. Its kcat is almost 1 million per second. Now that means each carbonic anhydrase enzyme converts 1 million molecules of CO2 plus 1 million molecules of water into 1 million molecules of carbonic acid every second. Carbon dioxide is poisonous at high concentrations, so having an enzyme converting it as fast as possible to something else makes good sense. But how is that possible? Enzymes provide the best examples of the difference between the macroscopic world we live in out here and the tiny world of molecules in solution. We cannot conceive of doing or making 1 million things per second. But in the submicroscopic world, molecules bop around extraordinarily rapidly. Consider that 1 million molecules of carbon dioxide and water must move into the active site of carbonic anhydrase in order to come out as 1 million carbonic acid molecules per second. For enzymes like this, the only limitation on its activity is the speed with which these substrates enter and bind the active site. The carbonic acid formed by the enzyme also plays an important role in our body, I should note. It ionizes to release a proton and form an ion called bicarbonate. Now, bicarbonate is important because it's a buffer that helps hold the blood pH in a very narrow range, as we've talked about before. Now, there's a downside to such speed as I've been describing. The extremely rapid catalytic action of enzymes makes it harder to fine-tune production to changing demand. If the garment factory zips along at top speed, that was a pun, by the way, and demand for the product stops, the factory will end up with a lot of garments it can't sell. Similarly, synthesis of biomolecules by enzymes with high kcat values could result in more product than desired being made before control mechanisms turn down production. Consequently, most enzymes have not evolved to have kcat values as high as possible. The phrase as high as possible suggests that there's a maximum rate enzymes could achieve. There is. Carbonic anhydrase is an example of an enzyme that has reached its maximum possible rate. In fact, it is in a small category of enzymes we refer to as perfect. Now, perfect enzymes perform at the theoretical optima of enzyme activity, which shows us that even the fastest enzymes do have limits. Perfect enzymes also exemplify another concept that we were touching on when I talked about the snugness of fit of an enzyme for its substrate. Biochemists use the word affinity. Affinity is similar to what we observed in the previous lecture for myoglobin, which was capable of binding oxygen strongly even when oxygen concentrations were low. Enzymes with a high affinity for substrate will have significant amounts of it bound even when substrate concentrations are low. To obtain the affinity of an enzyme for its substrate, we first determine the maximum velocity, its Vmax, for the reaction. In our factory analogy, Vmax is maximum production. Affinity is assessed as the concentration of substrate required for the enzyme to achieve half the maximum velocity, or Vmax over 2. The name we use for the measurement of this affinity, or desire for substrate by the enzyme, is called Km, or the Michaelis constant. Now let's consider another enzyme catalyzing the same reaction at the same Vmax as I've been describing. I've superimposed the plot of the second enzyme on the original plot. Examining it in this way, we see that this enzyme has a higher need for substrate, reflected in the higher Km value. A higher substrate concentration is needed to get to half Vmax than in the previous case. Now, why does this enzyme need more substrate to achieve the same reaction velocity? It's because this second enzyme does not bind to substrate molecules as strongly when they interact. In other words, instead of binding to every substrate molecule it meets, it only grabs a fraction of them. So if the enzyme only binds, let's say, every other substrate molecule, it takes twice as many substrate molecules bumping into it to achieve the same reaction velocity as one that binds to every substrate molecule it meets. A higher Km value therefore reflects a higher need for substrate due to lower substrate affinity by the enzyme. So a perfect enzyme is more than one that is extraordinarily fast. Speed of catalysis is only one consideration for a perfect enzyme. Substrate affinity is the other. So a perfect enzyme, like carbonic anhydrase, has a very high turnover number, kcat, and a very low need for substrate, as reflected by a low Km. Perfect enzymes, interestingly, all have similar and very large values for the ratio kcat over Km. So you might be wondering, why is this ratio for all perfect enzymes so similar? Well, as Tolstoy famously said, happy families are all alike. And the main reason is that perfect enzymes all face the same unavoidable limitation: the rate at which the substrate can diffuse through water to the active site of the enzyme. Perfect enzymes are diffusion limited. Without the speed and affinity of enzymes, you'd have uncatalyzed chemistry, which would leave you waiting around for 78 million years to make nucleotides. It kind of puts a slow internet connection in perspective, right? How enzymes perform their magic makes me think of work. When I've got a job to do, it takes me a certain activation energy to overcome before I can get started. Once I get going, though, the more things they throw at me, the faster I seem to work, and yet there's still a limit. If I were perfect, I might be able to handle everything as fast as it comes, but I'm not. I guess my affinity for my work isn't as strong as it could be. That said, the next lecture is all about what my boss tells me to do, or not do. Biochemists call it enzyme regulation.